In today's video, we're going to be taking a look at the difference between transcription and translation. So we have this process where we get a protein from a DNA, and throughout this process, transcription and translation occur. Now, to know at which phase they occur, I simply like to remember the definition of transcription and translation. Now, to transcribe means to change something from the audio format, for example, to the written format, but keeping it in the same language. So that's very important. To transcribe, we're keeping it in the same language. To translate, on the other hand, means to change something from one language to another. Okay, we're having different languages. Now, with that in mind, let's take a look at this process. So we have deoxyribonucleic acid, and then we get messenger ribonucleic acid. Both of them are nucleic acids. They're the same type, right? They're both nucleic acids. So we know that that process is going to be transcription. We then have messenger ribonucleic acid, and we get a protein, which is an amino acid, right? It's made of amino acids, sorry. So they're both different types. That we have nucleic acid, and then we have amino acid. They're different types, different languages, right? So we know that this is going to be translation. So we have transcription and then translation. Now let's look at the processes. Transcription has three different stages. The first is initiation, where an enzyme called RNA polymerase attaches to the DNA at a promoter region, just before the sequence of DNA that needs to be transcribed. The DNA double helix then unwinds, and the hydrogen bonds between the complementary DNA base pairs breaks down. The RNA polymerase, so after initiation, we then have elongation, where activated RNA nucleotides from the nucleus come and form temporary hydrogen bonds with their complementary DNA nucleotides. Now, it's important to understand that RNA does not have thymine, and instead it has uracil. So if we have a DNA nucleotide, which is adenine, right? If we have an adenine DNA nucleotide, the complementary RNA nucleotide that will come will be of uracil. So with that in mind, what then happens is the RNA polymerase moves along, and it creates phosphodiester bonds between the adjacent RNA nucleotides, which is recognized by this highlighted region, right? This highlighted region is to recognize the phosphodiester bonds formed. Now, as the RNA polymerase moves along, the DNA double helix behind it, well, the DNA behind it coils again into a double helix, releasing the RNA nucleotides. And this results in only about 12 base pairs being exposed at any one time. After elongation, we then have termination, where the RNA polymerase reaches a termination sequence, causing it to detach, and the RNA single strand to detach also. Now, we up here have our non-transcribed strand, and down here we have our template strand, or transcribed strand. So now we have a RNA strand. We need messenger RNA. So what happens is post-transcriptional modification. And what happens here is a guanine cap is added at the 5' end. So that's a single guanine nucleotide, and that is going to be used to set off translation. An adenine cap of 100 or so adenine nucleotides is added to the 3' end. And in DNA, we have sequences of DNA that don't code for a polypeptide, and they're called introns. And in post-transcriptional modification, those non-coding sequences of DNA, well, those non-coding sequences now of mRNA, so the non-coding sequences, are removed, and only the coding sequences, so the sequences that code for a polypeptide, are left, and they're called exons, and they're joined together. So our final mRNA will have a guanine cap, so one guanine nucleotide at the 5' end, exons, which will code for a polypeptide, and then an adenine tail of about 100 or so adenine nucleotides at the 3' end. Now, translation, okay, so we have our mRNA strand and we want a protein. Now, what are proteins composed of? They're composed of amino acids. So one of the first steps of translation is amino acid activation, where the amino acids form an intermediate with ATP, and then they attach to the amino acid attachment site of a tRNA molecule, forming an amino acyl tRNA complex. This here is a ribosome. We have a large subunit and we have a small subunit. The A indicates the amino acyl site. This is where the tRNA molecules will enter. The P indicates the peptidyl site. This is where the peptide bonds will form. And the E indicates the exit site. This is where the tRNA molecules will leave the ribosome. Now, before we go any further, it's important to understand that amino acids are coded for by triplets of bases called codons. Okay, so with that in mind, let's go ahead and look at the process of translation. So, the small subunit will attach to the 5' end of our mRNA strand, right at that guanine nucleotide. The large subunit will then come together to keep in place the mRNA strand. What will then happen is the ribosome will begin moving along the mRNA strand, and it will expose the first mRNA codon, which is the start codon, okay, which could be, for example, AUG. Now, with that first codon exposed, the tRNA molecule that has a complementary anticodon will come and form temporary hydrogen bonds, bringing with it the amino acid. Okay, the ribosome will then move along further on the mRNA strand, and then the next codon will be exposed, which will allow for the next tRNA molecule with the complementary anticodon to come into and form temporary hydrogen bonds, bringing with it its amino acid. And what that is going to do is it's going to bring the amino acids close to one another, and by means of an enzyme, a peptide bond will be formed. The ribosome will then continue moving along the mRNA strand, and the next codon on the mRNA strand will be exposed. Then what will happen is the next tRNA molecule with a complementary anticodon will come into the ribosome and form temporary hydrogen bonds with the codon of the mRNA strand, again bringing its amino acid close to the other two that were already that a peptide bond was already formed between, and then an enzyme will once again form another peptide bond. Then the ribosome will move again, and then once it moves, right, so if we had our first tRNA molecule, it moved along. The second tRNA molecule came in, the peptide bond was formed, the ribosome moved to the third tRNA molecule. That initial tRNA molecule is kicked out, right? It leaves via the exit site. So the most tRNA molecules that can be bound in the ribosome, right, bound to the mRNA strand, is 2. Now, what happens, so after the first ribosome attaches to the mRNA strand and moves along, up to 50 different ribosomes can attach afterwards to the mRNA strand. And when something like this occurs, it's called a polysome. And many polypeptides, right, many of the same polypeptides will therefore be synthesized, right? If we have 50 ribosomes working on the same mRNA strand, one after the other, well, we're going to have 50 polypeptides that are the same being produced. Now, the ribosome will continue doing this process until it reaches a stop codon, where the ribosome will detach and the polypeptide will be released, and we will have the polypeptide synthesized. Now, that's really it for this video. So the main one thing that I quickly want to reiterate, on the tRNA molecule we have an anticodon, and on the mRNA strand we have the codon. So that's it for this video. If you found it helpful, take a look at some of the other content on my channel that might interest you. Otherwise, I appreciate you taking the time to watch this video. I hope it helped, and have a great day.